When food is ingested, it travels to the small intestine where it is absorbed using the villi and microvilli, which are capillaries. This transfers nutrients into the bloodstream and to the hepatic portal vein. From the vein, it goes into the liver, where it will either be used for energy, stored as glycogen, or transported to tissues through the bloodstream, where it can enter the cells through the help of insulin or glut transporters. The insulin will bind to the receptors, which signal transductions, cascade, and exocytosis will occur, which is the movement of materials out of the cell. If the enzyme is denatured by way of heat or a change in pH, it will no longer function properly. When the enzyme sucrase acts on sucrose by way of hydrolysis, it produces two different molecules. Sucrase is the only enzyme that will break sucrose due to the structure-function rule.
Here we have the equation of cellular respiration. C6H12O6 plus 6 O2s is the product of 6 CO2 plus 6 H2O plus ATP plus heat. These are the reactants and the products of cellular respiration. The hydrogen of glucose is oxidized to 6 carbon dioxides, and the oxygen is reduced to 6 H2Os as a product. Here we have ATP as a product of the cellular respiration equation. Here you can see the structure, which is a phosphate group, a ribose sugar, and an adenine group, which is a nitrogenous base. The function is used as the main energy source for metabolic functions. They are consumed by energy requiring endothermic process and produced by energy releasing exothermic processes in the cell, which occur in the mitochondria. It does this by either losing or gaining a phosphate group through the phosphorylation.
There are two types of energy. An energy is the capacity to cause change or do work. The first is kinetic energy, which is the energy of motion. Within that, there's thermal and light energy. Thermal energy is associated with the random movement of atoms. Light energy is the type of kinetic energy that can be harnessed to power photosynthesis. Potential energy, the second kind of energy, is that matter possesses as a result of its location. Chemical energy is a type of potential energy, available for release in a chemical reaction. It is the most important in living organisms to power the cell. Chemical reactions either release energy or require an input of energy, which is an exergonic reaction or an endergonic reaction. For exergonic reactions, potential energy of reactants is higher than the potential energy of its products. In endergonic reactions, potential energy of its products is higher than that of its reactants. Energy coupling uses energy released from exergonic reactions to drive endergonic reactions. An example of energy coupling is the phosphorylation of ADP plus a phosphate group to create the ATP, which is an exergonic reaction. Hydrolysis is the losing of that phosphate group and releasing energy as an exergonic reaction. Phosphorylation needs the energy from hydrolysis to make ATP, and vice versa. Without one, the other does not have the energy required to function. Glycolysis begins cellular respiration by breaking down glucose into a 3-carbon compound called pyruvate. There are 2 phases in glycolysis. The first is called the energy investment phase, where you input ATP and glucose becomes phosphorylated to glucose 6-phosphate. Further down, the molecules are split into glyceraldehyde 3-phosphate. Phase 2, or the energy payoff phase, is where NAD plus reduces to 2-NADH. Also, ATP and pyruvate molecules are produced.
Glucose is energized using ATP. Glucose is a 6-carbon molecule, and right here we have our ATP. Here we're going to explain how glycolysis works. We are in the energy investment phase. The glucose is energized by using the ATP. A sequence of 3 chemical reactions converts glucose to a molecule of intermediate fructose 1,6-biphosphate. The cell invests 2 ATP molecules at step 1 and at step 3 to produce an energized, more reactive molecule. So here is our carbon moving down. Our ATP loses 1 phosphate group and it comes to our glucose 6-phosphate. And then the 6-carbon intermediate splits into 2 3-carbon intermediates. The enzyme splits the fructose 1,6-biphosphate into 2 3-carbon molecules of a glyceraldehyde 3-phosphate, or G3P. The 2 G3P molecules produced from each glucose enter into step 5. So now they both have their phosphate group. They're split into the G3P. A redox reaction generates an NAD or NAD to NAD plus DH plus. The pyruvate oxidation. It takes place in the mitochondria. 2 molecules of pyruvate are oxidized to acetyl-CoA in preparation of the citric acid cycle. The net molecular products are 2 CO2, 2 NADH, and 2 acetyl-CoA. There are no ATP produced in the pyruvate oxidation. This is the only process in cellular respiration that no ATP is produced. NADH donate electrons to the electron transport chain and then are oxidized to NAD+. A 3-carbon molecule of pyruvate enters through pyruvate oxidation. The carboxyl group is released as CO2 in the first step. The 2-carbon molecule remaining is oxidized and a compound called coenzyme A joins the 2-carbon group to form the acetyl-CoA. While this is happening, a NAD plus carrier is reduced to NADH, which carries the electrons to the electron transport chain to eventually be made into NAD plus again. Acetyl-CoA enters the citric acid cycle. This is where the coenzyme A is stripped away from the 2-carbon molecule that it attached to. It moves down the cycle and attaches to 4 carbons to make a citrate molecule, hence the name citric acid cycle. An NAD plus is then reduced to NADH plus another hydrogen. Our 6-carbon molecule moves down as CO2 leaves the cycle. We are left with 5 carbons. We keep moving and then we lose another CO2, so we're left with our 4-carbon molecule. We have another NADH plus that is then reduced to NADH plus another hydrogen. We then have an ADP plus a phosphate group that is phosphorylized to ATP. Our 4-carbon molecule keeps moving. We then have FAD, which is then reduced to FADH2. And this is completely exclusive to the citric acid cycle. Our 4-carbon molecule keeps moving, and then we have another NAD plus that is then reduced again to NADH plus another hydrogen ion. We then finally come to the top of our citric acid cycle to make oxaloate acetate, and with this our citric acid cycle turns twice, resulting in 4 total ATP, 10 NADH, and 2 FADH2.